Hi, everybody. Okay, so let's talk about meiosis. Um, Humans have unique versions of traits that cause us to look and act differently from one another. I want you to take a look at the people around you. Are they all the same? Aside from identical siblings, such as identical twins or triplets, there's a huge variety in physical traits from one person to the next. Okay, we don't all look the same. What do you think accounts for such a large amount of genetic variation within the human population? Take a moment, answer the question, and then you can move to the next slide. So, many different combinations of traits can occur when sex cells combine. This relates to variation in human traits, and that gives rise to how siblings look different, and parents and children look different, and the whole population looks different. DNA is genetic material in organisms. It codes for proteins and determines when proteins are made. A chromosome is one long continuous thread of DNA that consists of many genes. Genes then code for proteins. Humans have 46 chromosomes in each body cell. If the DNA in one cell was stretched out straight and measured, it would be 2 meters long. Each species has their own number of chromosomes per cell. Yeast has 32 chromosomes in 16 pairs. Ferns have 1,200 chromosomes, or actually more than 1,200 chromosomes, which is actually a world record. Humans have 46 chromosomes in 23 pairs. Homologous chromosomes are two chromosomes that you get one from each parent that have the same set of genes. Okay, each gene is represented by a colored band, like the picture on the right. They are the same gene, but the version that you have from each parent might be different. So, for example, if you have a parent with red hair and a parent with brown hair, then you're going to have like the same colored band, the same gene for hair color, but one of those is going to be red and the other one's going to be brown. Okay. Now, each chromosome pair is called a homologous pair. The chromosomes in all of your cells are genetically identical to each other, unless there's some kind of mutation, typically, okay, that's occurred. Now, during cell division, the homologous chromosomes are duplicated. Okay, each half of the duplicated chromosome is a chromatid. So the blue singular one right here, that's a chromatid. The green one, that's a chromatid. Together, they make sister chromatids. Okay, the sister chromatids are attached by the centromere, which is the center point in the middle of the X. Chromatids are copies of each other. Okay, they're really just they're exact copies of each other. Homologous pairs do not attach to each other. So here we have homologous chromosomes, right? We've got the chromatid. When it duplicates, we get the X shape. Okay, they are sister chromatids, and that's the copy. And then the two together, the blue and the green, those are homologous chromosomes. You got one from one parent, the other from the other parent. Okay, and so you have homologous pairs, which is the two pairs together. Okay, they do not attach to one another. So when you see these X-shaped chromosomes, those are sister chromatids, okay, not a homologous pair. And it's really important that you understand the difference between the two. Now, why do you think DNA is duplicated before a cell divides? Why do you think the sister chromatids are attached to one another? Well, it's because duplication gives each cell a complete set of chromosomes. Sister chromatids are attached at the center so they line up properly and divide evenly, and they don't get mixed up. Now, karyotypes are these maps. They let scientists study an individual's chromosome pairs. It shows each homologous pair in a cell. The chromosomes are stained with chemicals, and that gives these bands on them. The sizes and locations for those bands are pretty consistent across the chromosomes, but the bands differ. Karyotypes can show if someone has too many chromosomes or not enough, for example. Down syndrome is when you have an extra copy of chromosome 21. So that is this one right here. Okay. Either you have an extra copy of a whole or part of a chromosome. XYY syndrome is when you have a male presenting person that has an extra Y chromosome. So they've got an X, they've got a Y, and then they've got another Y. Okay, so here in the XY zones, they would have one more Y. Now, autosomes are the chromosome pairs for pairs 1 through 22. Okay, so 1, 2, 3, all the way to 22, those are all autosomes. These are genes that code for characteristics that are not directly related to the sex of an organism. They are ordered by size. 1 is the largest chromosome and 22 is the smallest. Now, sex chromosomes is the last pair. It's pair 23. In sexually reproducing species like humans, they directly control the development of sexual characteristics. In most mammals, sex is determined by the XY system. XX is biologically female, XY is biologically male. That's generally what we go for. Mosaic genetics, where we have are basically people who have a mixture of cells. They have some cells that are XX and some cells that are XY, and these people are on the intersex spectrum. XXY is when you have somebody who's usually assigned male at birth. Okay, they appear male, they have male genitalia or genitalia that's associated with males. This is called Klinefelter syndrome, but they have an extra X chromosome in their genetics. So they might have some female characteristics or they grow differently than the typical male. XX male syndrome is somebody who is intersex. So they have male sex characteristics like growth, like male growth hair, for example. They've got facial hair or extra muscles, or, they don't develop breasts, for example. And in this case, one of the X chromosomes has Y chromosome genes. So there was some kind of weird crossing over thing that happened. And one of those X chromosomes got genes from the Y chromosome. So they're genetically XX, but they present with male sex characteristics. XY gonadal dysgenesis is also an intersex person. This person would have female sex characteristics while also having testes. So their testes are usually underdeveloped, they're usually inside their body, and they would have female sex characteristics like a vagina, or a breast, but they would not be able to reproduce biologically, that is. Okay. So there are, and this is only a very small subsection of the many different possibilities that we can have in our XY system. Now, though X and Y pair with each other, they are not homologous chromosomes. X is a larger chromosome. It has a lot of genes, including ones that are unrelated to sex characteristics. Y, however, is just genes for testes and other male traits, or what we consider to be traditionally male biological traits. It is one of the smallest chromosomes, or the smallest chromosome, and it has very few genes on it. Androgen insensitivity syndrome, by the way, or AIS, is a person who actually genetically is XY, but develops female presenting genitals. And so that person would appear female and may even be categorized as female at birth or assigned female at birth, but is in fact genetically male or XY. And, that is in this zone of intersex as well. Now, what questions do you have about the relationship between chromosomes and DNA in terms of how traits are inherited? I want you to list one question. You need at least one to get a point for this slide. So come up with a question related to chromosomes and DNA, how traits are shared or inherited. What do you think? And your answers may vary. Now, body or somatic cells are diploid cells. They have 2 copies of each chromosome. These are not exact copies. You get 1 chromosome from each of your parents. In humans, diploid cells have 46 chromosomes, or 2N. Germ cells are cells that are found in reproductive organs. They make up gametes, or sex cells. So germ cells become gametes. Now, cells that are formed in the testes are called sperm. Cells that are formed in the ovaries are called eggs. Gametes only have 1 copy of each chromosome, which are haploid cells, okay? And that means they have 23 chromosomes, not 46. We represent this as N, not 2N. Now, each of the egg or sperm cells has 22 autosomes and 1 sex chromosome. Eggs are always X. Sperm can be either X or Y. So that is often why the parent with the sperm is the parent that decides the sex of the future offspring, typically. Why is it important for gametes to be haploid instead of diploid? What do you think? So it's because that gametes must have half a set of DNA. So when they fuse together, they make 1 full set. If you had a full set to start with, then when those two fused, the zygote would have two sets of DNA, and that's way too much. So polyploidy is when an organism has more than one chromosome. Okay, so when two gametes join together, we have this process called fertilization. Nuclei of an egg and a sperm fuse together, they make one nucleus. Now, maintaining that correct number of chromosomes is really important to survival of the organism. Changing that number is usually harmful, usually has negative effects. Increasing the number of chromosomes can, on occasion, lead to a new species. This is mostly in plants. It's very rare in animals. You can see some examples on the left of plants that have more than two sex chromosomes. Or, sorry, not more than two sex chromosomes, more than the usual amount of chromosomes. Germ cells go through meiosis to make gametes. Okay, meiosis is a form of cell division that takes one diploid cell and divides it into four haploid cells. There are two rounds of division. There's meiosis 1 and meiosis 2. They're the same stages as mitosis, except they happen twice. Okay, there's prophase, metaphase, anaphase, and telophase. It's really, as long as you can remember mitosis, you can remember meiosis. It's just a copy of it. Okay, it's two times instead of once. So meiosis divides one cell into four cells, but the resulting cells have half the amount of DNA as compared to the original cell. How do you think this is possible? Well, it's B. The DNA is copied once before cell division, and then when it goes through the second cell division, there's no copying. And so you go from double to normal amount to half. Now, what might happen if the DNA is not divided evenly between the cells? The resulting cell would have too many or too few chromosomes. Now remember, homologous chromosomes and sister chromatids are different things. Homologous chromosomes are two pairs, right? They divide in meiosis 1, the first stage. Sister chromatids split and they separate into new gametes during meiosis 2. So sister chromatids are copies of the exact same chromosome, and each one goes to a cell in meiosis 2. Now, centrosomes are those center zones that hold the sister chromatids together. Centrosome is a section in the cell that helps with cell division. I apologize, I'm getting confused. Centrosomes produce microtubules. They make centrioles. Okay, before cell division, the centrosome and the centrioles double. Then two new centrosomes move to opposite ends of the cell, and microtubules grow from the centrosomes and attach to the DNA at the centromeres, which then helps it divide. Now, before meiosis begins, a cell has to make a copy of DNA. Meiosis 1 separates the two homologous chromosomes, okay? So you get two haploid cells with duplicated chromosomes. So if you look here in this picture, you've got prophase 1, right? During this first phase of meiosis, the membrane breaks down, the centrosomes and centrioles do their thing, and those duplicated chromosomes condense. The homologous chromosomes begin to pair up. Okay, so remember, the homologous chromosomes are one from one parent, the other from the other parent. Now, they pair up precisely, gene for gene, down their length. And the sex chromosomes pair with each other. Some regions of the DNA begin to line up. Now, metaphase 1 is when the homologous chromosome pairs randomly line up along the middle of the cell by the spindle fibers, okay? And then the chromosomes, some from one parent, some from the other, are lined up along the equator, and this arrangement then basically mixes up the chromosomal combinations and really creates those genetic differences between parents and children. Then we have anaphase 1, where they separate from each other, so the pairs go to either end of the cell, and the sister chromatids remain together during this step and throughout meiosis 1. Then we have telophase 1, where the cells split apart, and we get two daughter cells. Now, the products of meiosis 1 are two cells with different combinations of chromosomes. One advantage is that we see the phases of meiosis 1 very clearly. The disadvantage is that we do not show the full set of 46 chromosomes found in a human body cell. Okay. Now, meiosis 2 separates those sister chromatids. The cells with chromosomes are not doubled, okay, during this stage. So we do not copy between meiosis 1 and meiosis 2, all right? So the end result of this is four haploid cells. So prophase 2, and this, by the way, on either side of the arrow is the 2 lines. So those are the 2 cells that were created that are now splitting into 4. We got prophase 2, where the membrane breaks down, centrosomes move to the opposite sides, blah, 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 blah. The spindle fibers align the 23 chromosomes at the cell equator. They go straight down. Each chromosome has 2 sister chromatids at this time, okay? So remember, the sister chromatids are identical to each other. So they line up, and then the sister chromatids are pulled apart. So now there is 1 copy of each chromosome, not 2, in each of the cells, okay? And they move to either side, and then we create 4 cells, all right? Now, what questions do you have about how chromosomes are sorted during meiosis? I want you to go ahead and take a moment and come up with at least one question. So remember, one question. I'm not giving you an answer, so you got to write a question yourself. The haploid cells made in meiosis cannot fertilize until they go through additional changes to make mature gametes. So they have to be mature. They can't just be, oh, we just meiosis and we're done. Nope. This process of making mature gametes is called gametogenesis. It is different in each sex. So, biologically female organisms have one way of doing it, biologically male organisms have another way of doing it. So, I want you to use the model that's on the left to infer which descriptions are accurately compare and contrast the production of sperm and egg cells in gametogenesis. So, which ones are true? And it's A and D. Both sperm and egg production involve meiosis, and both sperm and egg production begin with diploid cells. Okay, none of the others are correct. So eggs are created before birth inside the developing embryo's ovaries. So a XX organism in human would start developing as an embryo inside their, their XX parent's body, and they would begin developing, and only one of those haploid cells would make an egg. Okay, after the meiosis part, right? You got 4 of them. Only one of those makes an egg. The rest are polar bodies. Okay, most of a zygote's cell structures come from that egg. So when that egg then meets a sperm and makes an organism, that new organism, they're getting most of their their stuff from their XX parent, or typically their mother. Okay, mitochondrial DNA is passed from egg to embryo. Okay, so we pass it on from basically typically the biologically female parent would pass down their mitochondrial DNA to their offspring, because the sperm cells, on the other hand, really only pass down DNA. That's it. Okay, sperm passes DNA, and that's it. The eggs really do all of the heavy lifting in terms of everything else that the zygote needs. Sperm cells are created after puberty in the testes. Sperm needs to swim to fertilize an egg, so it has to move fast. So there are a lot of adaptations that sperm has developed to do that. The cell body is streamlined, so it's pretty much just DNA packed tightly in the head of that sperm. The flagellum is a tail; it acts as the propeller, and then the neck region that's the zone between the flagellum and the cell body, has mitochondria, and that fuels the flagellum. It gives it all the energy it needs to make the journey from the male testes to the female egg, basically. Meiosis and genetic variation. So meiosis and sexual reproduction lead to an increased genetic diversity in a species. Genetic variation is the differences in the genetic material of individuals in the population. Gametes have different combinations of genes than their parents, thanks to these things that happen in meiosis called independent assortment and crossing over. When homologous chromosomes pair up in metaphase 1, your parents' chromosomes line up randomly on either side of the equator, right? The arrangement of one pair doesn't affect the other pair, and this whole process is called independent assortment. We can calculate that. I'm going to let that go. It's very quiet. So basically, we've got this lining up, right? And they line up right down the center, and then they separate during meiosis 1. Okay. And we end up with the 2 separated chromosomes, or 2 separated cells. Now, to calculate the combinations of the possible chromosome combinations, we basically put 2 to the power of the number of chromosome pairs. So in this one, there's 3 different pairs. So we would have 8 possible combinations, and that's what it's showing in this video. So what is the approximate number of combinations in human cells? Remember, we have 23 different chromosomes. So we have 2 to the 23rd power, which is 8,400,000 combinations. Crossing over is the exchange of chromosome segments between homologous chromosomes. This happens during prophase 1, and it's a very regulated process. So we can see it happen in this video right here. The chromosomes and everything are preparing to divide. Each chromosome has already been duplicated at this point, right? The sister chromatids are still connected. We've got the homologous chromosomes that are paired up, and we get this weird thing happening here where the 2 chromatids touch each other and they exchange pieces, okay? Parts of one chromatid breaks off, reattaches to the other chromosome, and this can happen many times between the same pair of homologous chromosomes. Okay, it's another source of genetic recombination in cells. And so from now on, those now have traded that information, and then that gets passed to the offspring. How does crossing over contribute to genetic diversity? Well, it makes new combinations of genes from the parents, making the offspring more unique individuals. Once mature gametes have formed, they are ready for fertilization. This is when 2 gametes of different types fuse, like an egg and a sperm cell, to make a zygote. A zygote is a new organism, okay, with a complete set of DNA. They're getting half from each parent. By mixing and matching during meiosis and fertilization, we get increased genetic variation. In humans, each sex cell has 2 to the 23rd power combinations, right? 8,400,000. So if an egg and sperm cells combine at random, the total number of possible combinations is when we multiply the 2 together. So we get 8.4 million times each other. We get 70 trillion combinations. You are one of 70 trillion combinations that could have been possible to make up you. Now, in metaphase 1, homologous chromosomes exchange DNA segments in crossing over. Okay, this is where we get our genetic variation. Now, sometimes the chromosomes do not align properly, which means the 2 segments might be different in size. One chromosome could have 2 copies of a gene or genes, and that's called gene duplication. Other chromosomes could be missing completely a gene or multiple genes, and that's gene deletion. This leads to mutations, which could be beneficial or good for an organism or lethal. It could kill them. Gene duplication has occurred many times over millions of years of evolution. Domesticated sunflowers have a duplicated gene that lengthens the plant's growing period. This actually happened long before indigenous Americans began breeding the plants. They just preferred that variety of sunflower, so they kept breeding it, and that's the one that we have today. Okay? And that is it for today's notes. So please go ahead and turn this in, submit, and I will see you in the next one. Thanks, guys.